and in the past to all of us today.
All right, thank you so much, Cinema. Yeah, it's actually a real pleasure that I can talk to you guys, because the aim of this talk was also a little bit to introduce me to the crowd here, since I'm fairly new. This summer I worked with Marriott Cruise Group, and I commuted every day down here until I got really tired of it. And so I'm really happy to be at the coast. I'm from Switzerland, and as you know, it's not Sweden, so we don't have an ocean there. Sweden has the Baltic Sea and the Nordic Sea, but Switzerland, we're in the middle of the Alps. And actually I had a different picture first, but I was like, no, you have seen Newport so many times, the harbor, so I'll bring you a picture of Switzerland. I know people online won't see that, but this is actually the town where I grew up in, right around like here. It's a very narrow mountain valley. We get a lot of shade because the sun always seems to be behind the mountains. But yeah, so being at the ocean is just as nice. I really enjoy it here. So I wanted to start this talk with this graphic, and I apologize for the quite fuzzy graphics. There's a lot of pictures about One Health online, but not a lot of them, I think, make it onto big screens. So the One Health term, I think, was coined by the Center of Disease Control and Prevention. And the paradigm is the concept that human, animal, and environmental health are very tightly interconnected. And a lot of different specialties and fields contribute to that one common goal of One Health. So when I started vet school, I thought I would be mainly operating in the green topic around animal health, but there's a lot of human health is also involved in veterinary medicine when it comes to disease control and prevention, regardless whether I was in large animal, which I practiced first, and then small animal. So I also like this picture because it really shows how much collaborative research opportunities there actually are. And for the first part of this talk, I want to talk about the field that I've marked with a star. A lot of things fit into this field, like the human-animal bond that can lead to better health, but also animal products that provide a great food source for us. And also in this field fit diseases that are transmitted between humans and animals. So diseases are spread between human and animals, or humans and animals, or animals and humans, is called zoonotic diseases. In fact, actually, mostly we're talking about diseases that are transmitted from the animals to the humans, but we can also infect animals with our diseases, but that is not talked so often about. So the spread can happen via different ways. So there's direct contact by touch, or by bites, or scratching. And there's transmission via indirect contact via aerosols, or water, or instruments, or vectors such as insects like fleas, mosquitoes, etc. And then one big part of zoonotic diseases are transmitted via the food. So this schematic is not perfect, though. I'm not sure if anyone sees it. Why? I tried to put it in there. So the aquatic animals are missing, but that's a big part too. So let's talk about zoonotic diseases. So there's these four major classes. They're viral zoonosis, fungal, parasitic, or bacterial zoonosis. Let's start with viral zoonosis. There are, to my knowledge, no viral zoonosis that can be passed to humans from fish or shellfish by direct or indirect contact. However, there are marine mammals marine mammals are thought to be the reservoir for influenza, and influenza is the virus when we contract the flu, that's influenza virus. Far more common is the transmission via seafood. And it's mostly actually seafood that has come in contact with sewage. So it's not, for me, it's not really necessarily an animal disease, but that's what it is coined. So like shellfish mostly filter-feeding mollusks. They accumulate a lot of viruses that are in the water, and we infect ourselves by eating those contaminated animals from stuff we actually put into the water. And so the most common viral zoonosis that's transmitted by seafood is symbolized by this picture. Does anyone know where I want to go with this? Norovirus, right? Cruise ship curse. Maybe you heard of that. So, I want to say, norovirus is the most common one. Hepatitis A is also a risk. It's a lot more rare though. And yeah, the kind of like tragic thing is that norovirus and hepatitis A virus, humans are both the reservoir for it. So we're kind of like putting it back into the water. Norovirus outbreaks are such a big public health concern that the CDC has an entire section of their homepage dedicated to it. And so you can look at every single US cruise ship and can look at their inspection scores and reports, which is kind of interesting. So if you're planning to go on a cruise over Christmas, check it out first. I've copy-pasted here the cruise ship that got perfect scores in the last 12 months. It's actually not a lot of them because there's hundreds of boats.
Perfect meaning good or bad?
Good, perfect score. So 100, yeah, 100 is a good score. This ship actually, it's the one that has the worst score recently. It got tested, I think, the report came out in September. So that ship cruises in Mexico and in Alaska and try to avoid it. It has a lot of violations. The entire list, you can read through it. Very interesting.
I can't read the name on that ship.
Yeah, just check it out. I had it, I forgot, but yeah, it's a smaller shape, but yeah, and it's very expensive, so that's kind of interesting. All right, let's move on to fungal zoonosis. So again, to my knowledge, there are no fungal diseases that can be passed on from fish or shellfish, but again, marine mammals can transmit fungal diseases. And bottlenose dolphins are known to transmit not just one, but 3 different fungal diseases to humans. And one of them is lobomycosis. This disease is reported to have about a 7% prevalence in the Florida bottlenose population. So it's not a super common disease, but it seems to be somewhat emerging. It has been reported in Florida, also been reported in Texas and North Carolina. And it manifests in these proliferative like plaques that are all over the skin of the dolphin. And this is how lobomycosis can look if people contract it. So it builds these nodular skin lesions. And the cases that have been reported in the USA with lobomycosis are mostly from people that had contact with dolphins at aquariums or had seawater contact and had some kind of skin trauma or mosquito bites or just open skin lesions, as small they are, if you contract that.
Yeah. Are these unusual diseases, so are they curable, easily curable? Are they
Good question.
unusual to be curable?
Yeah, a good question. I would say it's probably, I'm not sure what the prevalence is of people infecting. Since only 7% of dolphins carry it, it's probably very rare, but it happens. This disease is not curable, so there's no drug, but its surgical is sizable. So yeah, with the ear, that might be a little bit bad news, but it is what it is. So, on the right side here, I have a photomicrograph of a biopsy of such a lesion, and on the right corner, you see these potato-shaped foamy cells. Those are macrophages. And if you look closely, I can show them here in the room. So there are all these spherical, pink, very round things that are yeasts. So lobomycosis is a yeast infection. And also very, I'm a pathologist, so I nerd out on these pictures. I really like them. The two big cells are called multinucleated giant cells, and that's very, I won't say it's diagnostic, but it's very special in fungal diseases because the the macrophages cannot deal with the fungal agents themselves, so they just accumulate other cells, but they essentially cannot deal with them. So next up, parasitic stenosis. So they are exclusively foodborne, to my knowledge. They're rarely reported, but there's a lot of papers say they're probably underreported, and that is because it's very hard to correlate symptoms with a parasite uptake via the food. So there's often a lag time, not so much with these guys. These are anisakis worm, and you can get symptoms within about 12 hours. But yeah, so they're not microscopic, so they should be easily seen. So that you accidentally ingest, I mean, it happens, but I think it's a fairly rare disease. But yeah, interesting. So Shuni and I went to the dock this summer, and there were guys filleting rockfish. And I'm not sure what I expected, but I was very surprised how many worms there were in those fillets. So there were like a lot. And so there's definitely around, very common in salmon and cod and rockfish. But parasites, they're very common in aquatic animals anyway. So parasites are really cool because they often have really cool life cycles. So the eggs are shed into the water, and then the larvae hatches, and the free-living larvae then gets taken up by small crustaceans. And the small crustacean gets eaten up by something bigger, and that fish or that squid gets eaten by something bigger again, so it moves up the food chain. If the parasite gets really lucky, it gets eaten by a marine mammal such as a whale or a dolphin, because only then the parasite can mate and reproduce, and the females can shed eggs again. If the parasite is unlucky and happens to be eaten by a human, it's kind of a dead end, so the worm will die in a few weeks, but not before giving you severe symptoms of really bad gastrointestinal disease, because it causes the parasite tries to bore through your gastrointestinal lining, and it just creates a really bad inflammatory response. So you probably know this. But the good news is all this is completely preventable. So not only is the worm can be fairly big, between 1/5 of an inch to 8 inches long, so you should actually be able to see it. But if you cook your meat, so those are the recommendations from the Food and Drug Administration. If you cook it to 145 degrees Fahrenheit, you're fine. And freezing is also an option. So in a regular freezing for 7 days, and the worms should be dead. If you have access to a colder freezer, the time will come down, but I don't want to recommend freezing meat in like a -80 or something. All right, and then to my favorite, bacteria are my favorite, so this is my favorite section. Contact transmission, like direct or indirect, is not that common with bacterial diseases, but I wanted to show the sea bass here because this seems to be an emerging disease in food fish. It has been in the past more of an ornamental fish disease. Anyone has an idea what that might be? I'll give you another picture. This is abdominal cavity. That big ragged organ is an enlarged spleen, and it has this white-gray granulomas in it. So whenever you see a granuloma in a fish, always think of the little sister of tuberculosis. This is mycobacteriosis. And so in fish, so Mycobacterium, they're very common in aquatic environments, in fresh and in salt water. In fish, it's mostly Mycobacterium marinum or fortuitum. And almost every fish contains some degree of Mycobacterium, so they very often have small granulomas. But since we don't necessarily eat kidney, liver, and spleen, we're usually good. So because that's the main organs. To have a skin manifestation like that, that's really a very severe case. But it is also an occupational hazard. So I'll show you this. So people who work a lot with specifically with ornamental fish. Handling fish or cleaning tanks, this has been a kind of like a common occurrence. It's called fish tank premalomus or fish handler's disease. Usually the bacteria get into small skin breaks, and then you get these nodules like this lady has on her third finger, on her middle finger. And then very characteristic for the disease is that those granulomas and ulcers, they occur along your lymphatic vessels, because again, the macrophages who try to take up the bacteria cannot deal with them, so they will transport them up in your lymphatic vessels towards a lymph node. And that's how that looks. This is treatable as well, but it can take up 12 months of antibiotic therapy, so they're very hardy. So if you contract the disease of aquatic animals, it will take patience. So lastly, seafoodborne, that's the more common transmission of bacterial diseases, almost exclusively from raw seafood or food items that were not thoroughly heated in a cooking process. So just because the clams, when you steam, they're open, it doesn't mean they're safe. Again, they have to be at internal temperature of, don't quote me on that, but probably also around 145 degrees Fahrenheit. Also, when we just talked about that before my talk, cold smoked products are also risky. Yeah, and so the most common bacterial genus that we can take up with seafood-borne transmissions is Vibrio. There's Salmonella, but then Salmonella and E. coli are again sewage-related, or people who prepare your food. So Vibrio are the main thing. And Vibrio, they're very common in the water. They have an extremely well-adapted ability to adapt to rapidly changing environmental conditions. So they can take very salty conditions to very low saline environments. And also they survive through a wide range of temperature. They really like warmer water, and if the water is warm, they grow in quicker than they already do. There are about 30 species known to date, and almost a third are human pathogens. So this is definitely quite a risk. The three main human pathogens are listed there. So Vibrio hemolyticus, it's the most common one. So if you eat raw oysters and have the unfortunate luck that they're contaminated, you get gastrointestinal disease. However, this is often self-limiting. If you contract Vibrio helmicticus, that's a lot more severe, but it's also a lot more rare. And then another species in that strain that's probably more known is Vibrio cholera, the diarrheal disease, the agent that causes the diarrheal disease cholera. And there's less in our waters because cholera really likes it a little warmer, but the southern states, you can actually get seafood that's contracted with or is contaminated with cholera as well. And then I also wanted to mention there's often like horror stories in newspapers about skin trauma. So if you have a tattoo, don't go into the water because Vibrio can really get you there. And then very importantly, Vibrios are also a very significant pathogen in marine aquaculture. And with that, I'd like to come back to this graph. And I find myself in the corner where the story is. That's where I like to do my kind of research. That's what I'm interested in. It's mostly sustainable alternative treatment options against aquaculture diseases in the areas of production, but also food safety. And as a veterinarian, I understand the needs for antibiotics, but still I had a somewhat eye-opening experience when I was in South Central Idaho and stood in a trout raceway of the largest trout producer in the world. And it's a really gorgeous environment. You have these canyon walls, and they have springs that come out of the canyon walls and cascade through the raceways. And at the end, they run through a settlement tank, but still they run directly into the river. And so some of the raceways were marked with red flags, which meant that these fish had gotten medicated feeds. And even though there's a settlement tank at the end, there's still no wastewater treatment. So residues of those antibiotics go straight in the river, and then downriver there are a lot of agriculture fields who use the water for irrigation, or even the water will probably also go in households. And that's definitely something that's dear to my heart, because I think we can do a lot better for that. That being said, after that experience in the raceways, that was in 2010 or 2011, I decided to study probiotics. So probiotics are nowadays, they're probably mostly known for being in yogurts, but they're nowadays in dried meat, in chocolate, in cereals, in skincare products. So it's kind of like a fancy new way, but like the poultry and the beef industry have been using probiotics for maybe the last 50 years. In aquaculture, we've been using it for the last 20. So it's nothing new. We also know that they work, but we don't really know why or how they work. And to start to understand how probiotics might work, we need to understand a little bit more the gastrointestinal microflora that is composed of a wide, diverse bacterial community, and this community balances each other. And there's often also pathogens or like disease agents in there, but through that balance, those pathogens are in check and they keep us essentially healthy. In humans, there are about 400 different bacterial species in the gastrointestinal tract, and they also have various roles. And some of those I wanted to show in this graphic. So the liver is the biggest digestive organs that we have. And the liver metabolizes proteins, fats, drugs, and also produces things like amino acids, or even bigger things like proteins, glycogen. And in order to get those things back into the bloodstream to be available in other tissues, they are conjugated to bile salts. And then they get, through the bile duct, they go into the intestine. And if we wouldn't have bacteria in that intestine, we wouldn't have the enzymes who would deconjugate these compounds. And so we can take up all those metabolites and those things that the liver prepared for us. So the bacteria have a very essential role there. Also, humans can naturally only produce vitamin B3, which is niacin, and vitamin D. All the vitamin A, most of the vitamin Bs, vitamin E, and vitamin K are all produced by bacteria. And then there's some, like vitamin C, that we have actually to take up via the food, because our bacteria cannot do that. But maybe at some point there will be a probiotic for that, I don't know. And also fiber, naturally we could not digest fibers, but we have bacteria for that. And then also, as already mentioned, these bacteria also take care of the first line of defense against pathogens. Oh yeah, so I also, where did I go? Yeah, so with probiotics, the idea is that we take advantage of these beneficial mechanisms and select them and use those and promote them as probiotics by giving them back in a higher amount or giving them to someone who doesn't have that function. So with that background, I started in 2011 a project that was part of my PhD thesis at Washington State University. I followed up on the work of David Burbank, who tested several probiotic candidates to be used against cold water disease, which is a fairly significant freshwater disease in salmonids. So I'm going to walk you through this. So he did a lot of work before that, but this is kind of like the last slide of his work. That's an infection trial, where he tested a bunch of different probiotic candidates that are on the right side of the y-axis. He fed those probiotics for 2 weeks before, on day 0, he infected the trout with the pathogen. And then over the course of 4 weeks, he kept continuing feeding the probiotics, but also watching out for mortalities. And the mortalities here are on the y-axis, so it's cumulative % mortality. And if you just look where I put the arrow, that's a positive control, so empty squares. The mortality, So these were fish that did not get any probiotics. There were 3 tanks of 25 fish each. And they had a mortality of approximately 50%. Then on the x-axis, the empty diamond, that's the mock infection control. So these fish did not get any probiotics either, but also did not get infected with the pathogen. They were just injected with a saline. So they had no mortalities. And then if you could only look at the one with the filled squares, that's the probiotic candidate C66. And that showed a significant reduction in mortality of about 50% compared to the positive control. So what I was most curious in my work is why we got that reduced mortality. And I wanted to look at it on what happens on the molecular level. So rather than using genomics, which I could have probably done just as well, I tapped a little bit into proteomics to reveal the mechanism of these probiotics. So the first step for me was to find out what the size was of that inhibiting compound that we knew was also in the growth media. So if you grow bacteria, you grow them in a growth media. And we found out that not only the bacteria or the probiotics itself have inhibiting function, but also if we took the bacteria out of that growth media, the growth media itself that didn't have any cells anymore retained an inhibiting activity. So we knew that there was something secreted from the probiotics. So, and I also knew that it was a protein because digestion would lose the effect. So for that, I just put the supernatant through a size fractionation filter and then tested the different fraction in a growth experiment against the pathogen. So on the y-axis is the average optical density of CSF259. That's just pathogen. And optical density is how we measure. So if the optical density is high, it just means the culture is very dense, and also means there's a lot more bacteria in there. And then all the way to the right, there's the control. So that's the pathogen grown without anything else. And then right next to it, to the left, there's the fraction that contained all the components that were larger as 3 kilodalton, which kilodalton, that's just a measure of molecular weight in proteins. And so if we added that fraction to the probiotic, if we added that fraction to the pathogen, nothing happened. The pathogen grew just as well as the control. But if I took the fraction that was smaller than 3 kilodalton, it had a severely inhibiting effect, just as much as the unfractionated supernatant from the probiotic. So I knew that was a very, very small protein. So 3 kilodalton is very small. So next up, I wanted to filter out that little peptide so I could put it to sequence analysis. So how I did that, I put it into an SDS page shell. The SDS page shell is a fibrous matrix where a current is applied, and the protein will be linearized, so denatured. So it's, instead of, usually proteins are like nicely folded up, so it will be linearized, straightened out. And the larger that element is, the slower it will go through those fibrous matrix. And the smaller it is, the quicker. So the 3 kilodalton band will be all the way to the bottom. So on the left side, you see that what we call a ladder, which are just it's a reference substance that will help us run our product at the same time and then see which sizes we're hard. And all the bands just mean there's a lot of stuff in there. We don't necessarily know what it is, but we know that it's like something proteinaceous from that side. And since I knew it was around 3 kilodalton, I simply excised that piece and submitted it to mass spec. And mass spec gives you an amino acid sequence that you can compare to a database, so you get some idea what kind of protein it has been. And I got a bunch of hits, and a few were like, oh, unspecified, or protein of unknown function. They were like, oh, great, right? I don't learn anything of those. But then I had one protein in there that was a toxin, antitoxin protein. I was like, wow, that sounds really cool. So we read up about it. So an antitoxin toxin system is like, it's like two proteins bodied together, but if they are expressed, the toxin kills all cells that are around them. The problem is it will also kill itself. So for that, it has an antitoxin expressed at the exact same time. And that antitoxin will prevent or protect the cell from killing itself by its own toxin. So I thought that's a really cool mechanism, and that could be possible in my mode of action of this probiotic. So next, I did not bring in the next slide, but what I then did was more like genomic stuff. So I created a genetic deletion, so what we call a knockout. So I just knocked out that gene in my probiotic. And to also, and then, test it on an agar plate, whether it's still inhibiting. But then sometimes you say, well, the knockout could have all kinds of different effects on the cell that's like rendering it useless. So what you also do, you create a complement. So a complement is basically a knockout where you bring in the gene that you deleted again in, and that should restore the phenotype. And then also what works in the lab doesn't necessarily need to work outside the lab, right? So I did the same trial that David Purbanks did. So I did an infection trial with my strains, which are here in that box on the right side. So again, I fed 3 tanks of 25 feet each who got one of the diets for 10 days. And then at day 0, I infected them with the pathogen. And then monitored survival over 4 weeks. And on top you see mock infection control. So this is a survival curve, I'm probably familiar with Kaplan-Meier, which starts with remaining live fish on the y-axis. So 25, I did the average. So 25 was my max. And the mock infection control was just saline injected fish. They did not die. And then the complemented strain in the wild type probiotic fed fish survived just fine in compared to the fish that received the probiotic where I had the gene knocked out, which died in similar numbers to the no probiotic control, which were fish that didn't receive any probiotic but were infected. So also gotta say with this project, I got really lucky that the mode of action was a toxic protein. Normally it might not be so easy, and I'm gonna show you why here in a bit. So shortly after that paper got published, I started working on a postdoc at OSU in 2015. And as a hobby on the side, I just started to screen marine bacteria because now I was finally closer to an ocean and I could do stuff like that. So I tested strains that I collected in seawater or oysters and shrimp and tested them whatever I could find in our freezer. So there were like human and as well aquaculture pathogens in there. And this picture shows one of the screens I did. And so how these are done, so these are agar plates. And as the first step, you spread your target pathogen on. I think this was Vibrio parahemolyticus. And then you drop your probiotic candidates or just the isolates that you have, drop them on little drops and let them grow. And then as you maybe can tell, the lower middle, there's this yellow strain. That's M1 that started all at OSU here. That shows this zone of inhibition around it that's pretty severe. Interesting, however, it doesn't inhibit the 4 strains around it, but it does inhibit the background. So they have species, they're very selective on what they inhibit. And then also on the right, there's this white strain right at the wall of the Petri dish. That's a bacillus that also has a little bit of an inhibiting zone around it. Now this is how I do the first screen for potential probiotics, and I know it looks really easy, and it kind of is, but then it also is not. So I'm gonna, but sometimes you get lucky, just like in a previous study, right? So I want to talk a little bit about, that's something I'm really interested in, what the mechanism of action of a probiotic actually is. So on that agar plate that I just showed you, that was a direct antagonism. And that could be either that the probiotic excretes something into the surrounding, and then you see it. But sometimes they do contact inhibition. So they only kill the body that's just next door, and then you won't see anything on the agar plate. And sometimes you also don't know whether they are just inhibiting the growth. So there's a difference between inhibiting someone or killing them. And then another mode of action could be that the probiotics simply outcompete the pathogen for a nutrient and starve them. Or the probiotic bioconverts something like a sugar into a fermentation product that is inhibiting itself. So it's like an indirect function. Or, as mentioned, bacteria produce vitamins, and these vitamins can be essential for either other bacteria or for metabolic functions in our body. And then I really like this function. Probiotics can just be a layer of protection on your gastrointestinal mucosa, and so prevent pathogens from actually entering, attaching to the epithelial cell of your mucosa, or even entering through the gastrointestinal barrier and going to the bloodstream. And what also could be a mechanism of action could be stimulation of your immune response. Or, and that's not on this graph here, bacteria could just be food and thus have a beneficial effect on the host because they provide fats and proteins. So going back here to another of those agar plates, so just to show that there's a wide variety of different mechanisms, but what we can only see very easily is those inhibition plates, and this is one against Vibrio vulnificus. There are a few other limitations too. I truly believe there's a cure for anything. Well, don't quote me on that, but there's a lot of cures in the ocean because there's a lot of bacteria. Unfortunately, we're only able to culture about 10%. So there's a lot of mystery and secret out there that we still need to discover. Yeah, but it's probably safe to say that most bacteria have some kind of mechanism that we could actually exploit for a beneficial effect. We just need to figure out how. And that brings me to the last few slides here. So since finding that M1 strain 4 years ago, myself and Chris Langdon have gotten probiotic work funded through National Sea Grant and NOAA. And the aim was to find probiotics beneficial to oyster larva culture that are often heavily impacted by another Vibrio species, Vibrio coriolis. So the aim was to improve the survival of the oyster larvae, to improve growth and settlement. And so these last 3 slides, he's not here, but David Burbanks did those, so I have to credit him. He did all the work for those. So for this work, we looked at adult oysters and wanted to screen their gastrointestinal environment, or at least water that was somehow close to oysters, because I assume that if an oyster is healthy, it probably has an abundance of beneficial strains in its gastrointestinal tract, and will probably naturally have defense mechanisms that can defeat those pathogens. So we then screened them on agar plates like this one I showed before, and also tested that we didn't use Vibrios as probiotics, which they also can show inhibiting activity against this Vibrio. And then David tested the probiotic candidates on small mortality assays by counting dead larvae after co-culturing the larvae with the pathogen Vibrio coriolis and also the probiotic candidate. And I can walk you through this slide real quick. So on the y-axis is % mortality. That's pretty straightforward. And then I know it's too small to see, but on the x-axis are different probiotic candidates. The ones that are blue, that's kind of like our first test. We need to find out whether the probiotic itself is a pathogen to oyster larvae, which that's something you don't know unless you test it. So that looked all fine. Where the green arrow is, that's a larval only control. So there's always a little bit of mortality with regular healthy larvae in those assays too. So they were, all the blue ones, they were in line with that. Now the red ones, those were the larvae that got cultured with Vibrio chlorhydricus and as well with a probiotic candidate. And I put the blue arrow in, this is the positive control. So it's larvae that are cultured with Vibrio chlorhydricus strain RE22. So fairly high, I think almost 100% mortality. Then the 3 strains on the right and the 2 strains on the left, they were really bad luck, so they did not inhibit or reduce mortality. But then these strains, they show a lot of potential because they reduce mortality at least by half. So we're somewhere in a mean average of 30% mortality compared to almost 100%. And then also David was measuring out all the larvae in there, and he found out that the larvae also grow a lot better. So the blue bar here, that's the larvae that did not receive any probiotics. But then the red and green, those are larvae that received at least 1 dose of probiotics, and they grew significantly better during the same time. This could be because the probiotics gave them some extra nutrients that they could grow faster, or that they relieved some kind of stress of the oyster larvae so they could put all their energy into growth instead of fighting with whatever around them. So we don't know yet at this point. And this is the last slide. I know probably not, so I just want to, well, since in the room I can show. So on the top are, these are marble tiles that David put into the water with oyster larvae. And when the oyster larvae is ready to go from the plankton stage to sessile stage, they will set on substrates, which what the marble tiles were for. So the ones on the top, the 2 rows, these were larvae that were never exposed to probiotics. And the bottom ones, they got 1 dose of probiotic 24 hours after being fertilized. And larvae settle, I think, after about 2 weeks or a little longer. So they only got those ones, and they had some kind of a health benefit memory during this time. And I hope you can appreciate that here all these gray patches, that's all oyster larvae that's set, and then compared to the top ones, there's really very few of them. So now we can speculate what these probiotics really do, but they do modify the microbial community in some way that's extremely beneficial to those oyster larvae. So there will be a lot of interesting things, because we also have a side study going with this where we look at the microbiome, so the microbial communities in those larvae, and see what changes with the probiotic addition. Yeah, but we're probably going to talk about this next time. And with that, I thank you very much. Open for questions. Yes.
I'm curious about quantity of probiotics you had, just like, I'm sorry, I know nothing about quantity of antibiotics too. Years ago, when I did microbiology, there was a zone where if you added too little, you increased the growth of the pathogen. Does that still happen? Can you add too little of something? We used to do these bonds on agar plates, and antibiotic down in the zone.
Yeah.
Where there was not enough, you increase the bacterial growth as though the antibiotic was a nutrient or something. You see that with probiotics?
Yes, dose dependency is one of those hurdles that sometimes you don't recognize a probiotic when there's one there. If you had too much pathogen, that just often gets overpowered. It's often a function of how fast the 2 species or the 2 strains grow. So yeah, you can totally miss the effect. Yeah, that's definitely still, I have not solved that yet. But then also, I mean, those agar plates, that's basic microbiology, that's really kind of easy to test, with the caveat that there's a lot of factors that we could just miss. But I assume in the next few years we'll do that with gene mining. We just look at databases of bacteria and then have prediction of what could be beneficial. The next hurdle, of course, then is like whether we can culture it or not, but we'll probably make progress there too. And then there's always the possibility that if you identify a molecule, for example, that's really important, you can scientifically engineer it, so you don't really need the bacteria. So I think there will be a lot of advances in probably the next 20, 25 years in that. There was a question.
Yeah, I just had a question. Probiotics, I don't really understand exactly what it is. Is it a general term for a lot of...
Oh, yeah. Good point. Yeah, I'm sorry, I totally missed that. So a probiotic, the World Health Organization defines a probiotic as a microorganism that provides a health benefit to the host. That could be whatever, so it's a very broad term. But it's just, there's good bacteria and there's bad bacteria, and probiotics are just the good ones, or at least the ones that don't not harm anything. So yeah, but in fact they're just bacteria, and we just take advantage of one of those metabolic ways or things that they do. So it's nothing really special, it's just a definition. But yeah, good question, sorry I forgot that. Yes.
I'm wondering, could there be any possibility of the fact of like dosing water with probiotics and then that changing the microbial community like on the settlement plates?
Yes.
Instead of within the larvae? And did you guys account for that or test for it?
Yeah, so that's another mode of action of a probiotic. A probiotic could just be a water, what's the term? A water conditioner, or yeah, improving the water, something in the water. That could be totally what works. Yeah, yeah. So we'll have to do some more research to find out. We try to swap the plates because maybe there's a biofilm that increases the settling, and so maybe in the biofilm there are probiotics that we haven't intended. So there's probably some interplay, but it's a microbial community, so they all talk to each other. So it's a big field. Yes.
And so in your previous work, when you saw that this probiotic was secreting like an antitoxin protein,
Yeah.
Why do you think it was doing that? Was it doing that because to protect itself from other bacteria that secrete toxins that it normally occurs with, or does it sometimes turn on a toxin and is protecting it's like a self-protection?
Yeah, so it was interesting that gene was only mentioned in the literature once, and they totally disregarded it as something completely not relevant because how they did their study gave them a different results that how I would interpret it. But basically I think it turns on with quorum sensing, and quorum sensing is how bacteria talk to each other. And that paper thought that if the amount of bacteria is high enough, then that bacteria will produce the toxin and kill everyone off to save someone else, because the nutrients will be exploited at some point. But I looked at, and I blanked out a bunch of my graphs, the half-set graph, because I was using an E. coli lab strain, and I was really disappointed that it was also inhibiting. And I was like, well, so our product is not that special. And so when I found the gene, I compared gene sequences, and I found out that this gene is highly conserved in evolution, so a lot of other bacteria have it. So it must be something that's really important that all these bacteria put a lot of effort in to keep it over the years. So yeah, unfortunately we never got more funding to really look into it more, but essentially since it's such a small peptide, you can produce it in a yeast and then use it commercially. And there's a spin-off from the University of Cornell who did that. So they're producing this peptide now, obviously with some good results in aquaculture. But yeah. Yeah.
Yeah, how much do the environmental factors affect like temperature or light conditions, or the presence of another bacteria affect the functions of the probiotic?
Probably a lot, yeah. So just with the example, so Vibrio is not a good probiotic by any means, but since Vibrios really like warmer water. I assume we will hear a lot more of them with ocean warming. And ocean warming will affect the entire microbial community. We might lose really good ones, and then we'll maybe have more really bad diseases coming from the Vibrio side, who knows. But yeah, conditions, we can only study the conditions as they are at our, ambient when we want, but environmental influences, and that's the One Health paradigm. So there's so many things that contributed that we need to look at it from all the different sides of the disciplines to really figure that out.
I'm thinking from the, for example, if you have a normal temperature, 96.8, 98.6, whatever it is, and what if you raise your temperature like to 102 and try it? Would that kind of change the whole calculus of what might be going on?
Yeah, it could be. I'm not sure our probiotic would survive a lot of warming. Yeah, we haven't tested that yet, but it definitely temperature changes the dynamics a lot, yes. All right. Thank you. Yeah, that question did not come. I still eat sushi.
Oh, do you?
I don't eat raw oysters.
All right, everyone online, we're going to end the presentation. Thanks so much.
Yeah, I use more of a tool.
Okay.
So you had to do
Oh, okay, cool. Yeah.